Welcome to the fourth day of the Computational and Experimental Chemistry Symposium. The symposium is dedicated to bringing together the computational simulation community and experimental researchers to exchange scientific ideas and to learn the current state-of-the-art modeling, analytical and numerical tools together with advanced experimental techniques. We hope that our attendees, colleagues and their families are safe in the ongoing COVID-19 pandemic. Today there will be one lecture presented by Dr. James McDonagh from IBM Research. I'm Dmitry Lepshin, a PhD candidate at the University of Edinburgh and initiator of this series of lectures. Questions to our speaker from Zoom and YouTube will be moderated by me. Please use chats to ask questions at any time. I would like to acknowledge technical support provided by the International Adsorption Society and Dr. Dancy Darius in particular. Now I would like to introduce our speaker, Dr. James McDonagh. He's a research staff member at IBM Research Europe. His research interests include chemical property prediction and design, coarse-grained force field development, and applications to chemical formulation. He contributed to novel methods for force field optimization utilizing Bayesian optimization. With this, I would like to give a word to Dr. James McDonagh.
Thank you, Dmitry. Can everyone hear this okay?
Yes, it works well.
Okay. And the screen is up?
Yes, the screen is good.
Perfect. So good evening, everyone. Thank you for coming to join this, this presentation this evening. And I hope that we're finding you all well, given the current circumstances. I'd also like to thank Dmitry and everyone else that's been involved in the organization. It's been really great. So I'm going to present to you today some of the work that I've been working on for a little while now, exploring phase diagrams, predicting boundaries using molecular simulation. Just to begin with, I'd just like to give an outline for this presentation. So I'll start off just laying out some of the background and setting the scene for this work. Move on to discuss briefly the simulation methods that we've used, and then finally give a couple of use cases. So just to set the scene and set up the motivation behind the research, I'm based in a UK research lab for IBM, and I work in a team that has pretty broad interests across chemistry all the way through to genetics and technology, including cloud computing and AI. The whole team's motivated by projects that are of academic and industrial interest. So so we're looking to interface between these two areas. The work today is from the chemistry materials area, and it's been motivated by the UK formulation area, which we've been working in for the past few years. We've been working as a collaboration with the Science and Technology Facilities Council, which is also based in the UK, on looking for ways that we can use computational methods to enhance formulation R&D. So the team from STFC is all based in the UK, along with several others from IBM. And we've also had inputs and technical direction from researchers in IBM based in Ireland and the USA. So the motivation then for this talk comes from the perspective of formulation chemistry. And when we began doing this work, we went and spoke to a number of people in academia and industry about the kinds of areas they were interested in. Many of them mentioned phase behavior as an area of interest. And critically, they weren't just interested in predicting a boundary, they also wanted to be able to develop insight. Having done this, we then moved on to set up a scientific project. and we focused on liquid phase diagrams for the most part. In order to gain the insight that was needed, as well as the predictions, we've utilized molecular simulations, as we felt that would give us a good balance between being able to provide insight and predictive capability. The approach that we've taken throughout this, and we've also applied to the formulation work more generally, is to build repeatable and reusable workflows. And by doing this, we can encode the current state of the art in terms of simulation and analysis, and we can also enhance the reproducibility of our results, as it's easy to rerun these workflows once they're created. The workflows are also highly tunable, so we can replace one simulation with a different type of simulation and one analysis with another one. So we can quite quickly build up to a sizable number of workflows that are able to do a few different things. Just to give a sort of high-level pictorial illustration of this, the automated and reusable workflows that I mentioned. So there's a diagram there at the bottom of this triangle, which actually works very well for this presentation. It looks like a ternary diagram, but the diagram just to the side at the base there shows a graph of one of these workflows as it was running. So it's taken a single user input, spawned a load of simulations and analyses, and eventually pulls the information together to provide an interpretable output. Excuse me. In the center there, you'll see a parameterization engine. This was a project that we actually span out from this work in order to develop potential parameters for specific simulations, namely dissipative particle dynamics. And I'll mention a little bit more about this in a few slides' time. Finally, the top layer, this is a consumable computing interface, which is a fancy way of saying a user interface. And this enables those that perhaps aren't so used to using the command line or computational interfaces of that nature to be able to access and utilize some of these workflows through a much friendlier application. For the rest of this presentation, I'm going to focus on the science behind the reusable workflows and briefly on the parameterization engine. Just before I get to that though, I'm going to just introduce the two simulation methods. So the first one of these would be molecular dynamics, which is now arguably the quintessential way of simulating chemicals in general. For those that are less familiar with this methodology, it's a classical simulation method. And by this, I mean that the dynamics and the motion of the molecules that are in the simulation are propagated using classical physics. The molecules themselves tend to be represented as explicit atomic structures. This represents each atom explicitly, typically. The methodology itself then calculates interactions using a set of potentials, which we term a force field. These potentials can be split into a few different terms, and one way to split this would be those that operate to represent intramolecular terms. These are within the molecule. So these are representing motions such as bond vibrations, angle bending, and rotational and dihedral torsions. Other terms in these potentials are those that represent larger or longer range interactions. And those would be terms such as van der Waals interactions and electrostatics using the Coulomb potential. In this work, all of the molecular dynamics is carried out using the OPLS force field, and the simulations are carried out using the LAMP simulation package. Second simulation method that's used in this work is that of dissipative particle dynamics. This is also a classical simulation methodology, and it's quite similar in a lot of ways to molecular dynamics, but it does have a key difference, which is the fact that this is a coarse-grained method. What do I mean by coarse-grained? Well, if we took the explicit atomic representation, then a coarse-grained structure would simply be if we took taking subsets of those atoms and considering them as a single site. What this does practically is it means that the number of interactions we need to compute are reduced, so it's reducing the dimensionality of the problem. As we do that, we can simulate larger and longer, so we can simulate bigger systems for longer time periods. potential itself in DPD has, again, some similarities to molecular dynamics. So there's, again, a bond and an angle term, which are represented on the left and in the center. Main difference here is that these bonds and angles are now not between atoms, but they're between beads. In addition to this, the main potential that introduces the chemistry into DPD is the so-called conservative potential. This potential has this prefactor, Aij, which you can see at the front of the right-hand side equation. And this represents how repulsive a bead of type I is to a bead of type J. By doing this, you end up with the relative repulsiveness between beads, and so those that are more favorable to be closer together will be, and those that are less favorable will be repelled. And this is how we encode the chemistry principally into the DPD potentials. There are no standardized potentials like there are for molecular dynamics within DPD, so the literature does have quite a wide range of different potential parameters that can be used. But typically these have been parameterized for a specific use case, or often they've been parameterized against chemical intuition representing expected mutual solubilities, for example. So in this work, we've had to parameterize some of our own potentials. All of the DPD simulations are carried out using DL-meso within the rest of this work. Okay, so this brings us to our first use case. So this first case is investigating a ternary diagram, and this is where we'll be considering the relative ratios between three different chemical components. In most of the projects that we've run in this area, we started from the same point, and that is to go and explore the experimental data in order to locate a suitable benchmark set that we can validate our simulations against. In a lot of the formulation related work, we've sometimes found this to be difficult, either because the data simply wasn't published, or because it's been very hard to validate the small amounts of data that were available. Within phase separation or phase diagram generation itself, actually we found that there was a lot of well-curated experimental data that had been well validated, and so we had some choices to make. We eventually made the choice to go with the water methanol toluene diagram. We did this because it's got some interest both academically and industrially. So all three components are often used as solvents, possibly as reagents as well, but particularly the use case for solvents. Solvent reuse is becoming is already an important part of industrial processing, and toluene actually forms an azeotope with methanol and water, which can make it quite difficult to separate these three components. So understanding the phase diagram has a value from that perspective. The molecules that we used are shown in the lower image on the right-hand side, and the DPD coarse graining is shown by those red spheres that are overlapping the tops of the structures. Okay, so now we have some experimental data. We now need to decide which simulations we're going to run. We decided to run both MD and DPD in this case, and to compare the outputs of the two. As I mentioned before, MD is run using LAMPS and DPD using DL-meso. In both cases, we've thermally equilibrated the systems in the MBT ensemble, and then we've used the MPT ensemble for our sampling. There's just one stumbling block before we could start, and that was that we didn't have all of the potential parameters that we needed for dissipative particle dynamics. As I mentioned, there's no off-the-shelf set of potentials parameters that we can use for DPD, and so this meant that we had to go and parameterize our own. So as we found that this was the case in a number of the different formulation projects that we were running, we actually span out a separate project looking specifically at the parameterization. We developed two different methods for doing this. The first is a local gradient descent method, and that's the one that we'll be discussing over the next couple of slides. And the second is a global optimization methodology that utilizes a machine learning method to have quite efficient sampling over a larger space. But we won't discuss that one in any detail here. The parameterization that we do is top-down. What I mean by that is that we're parameterizing against known experimental data. So we're looking to reproduce experimental data in this case. And the code that we use for this is a locally modified version of force balance, which has previously been used for parameterizing molecular dynamics force fields against quantum chemical data. So to be completely specific about what we're doing here, we're looking at the conservative potential, which I mentioned before, and we're parameterizing those Aij terms. So we're looking to bring some chemistry into these models by making sure that we can match the relative repulsiveness from that potential to some experimental macroscopic observable. The algorithm that we apply for the optimization itself is the well-known BFGS gradient descent algorithm, and the gradients here are describing the change in the observable property with respect to the parameters of the potentials themselves. In this case, we were parameterizing against experimental density values, and we were parameterizing specifically the methanol bead and also the methyl group on the toluene molecule. Just for an illustrative example here, we have two diagrams on the right-hand side. Top diagram represents the starting point where we've selected some parameters that we believe may have been suitable based off of previous knowledge of what the potentials were for other chemical groups that were similar. And that's represented by the orange curve. The blue curve represents the experimental data. And we can see in the top diagram, they're really not overlapping very well. We then run our parameterization methodology, and the final output is given in the lower diagram. And you can see now that the orange curve is much more closely aligned to the blue curve, and we're off by only a few percentage points in terms of the error between the different points. The final parameters then that we arrived at are given here. And so you can see there's the parameterization runs there for methanol and for the so-called BCH bead, which is the methyl group on the aromatic ring. The rest of the parameters were taken from a collaborator's previous work in Anderson et al., or utilizing the same methodology. And those were parameterized against partition coefficients, so log P, and density themselves. So this now held us in a pretty good point. We had our simulation methodologies, we had our experimental method, and we now had all of our potential parameters. What was left now was to start determining how we could go about understanding the process of phase separation and how we could probe that through the molecular simulations. So we paused here and had a think about exactly what we wanted to be able to understand here. And really, when we think about phase separation, what we're thinking about is the density of a given molecule or entity changing across the simulation. So we're looking for that density change. In order to probe this via the molecular simulations, we decided to break down the simulation cell. So on the diagram on the right, you can see we begin with the whole simulation cell. And then in the next image on the right, we've selected some sub-region of that cell that we're going to focus on. And we do this exhaustively until it covers the whole simulation cell. On the diagram below that, you can see that we've highlighted a molecule, so toluene in this case. We count up the number of toluene molecules, and we do this for each of the subcells that we've generated. And we calculate then a distribution which represents the change of that system, the change in that toluene density across the system. We do this across the whole simulation itself, so the whole trajectory that occurs from the simulation. By doing this, we then have a single distribution which can represent the entire simulation, and we can use this to begin to understand what's occurring within the molecular simulations themselves. So to put this into some more physical and sort of meaningful terms, we had to think about the signatures of phase separation and what we expected to see from that perspective. So the first one, somewhat obviously, is a mixed state. And here we were expecting molecules to largely diffuse and act independently And we expected to see approximately an equal number of molecules in each of the subcells. This is shown by a relatively unimodal distribution that's shown in the image below. And indeed, we can see these kinds of signals in the distributions that we generate. The second case was as we begin to approach the phase boundary. And in this case, we would expect that molecules would start to act in a more correlated fashion, and we would be ending up with small clusters acting as units. When this occurs, we start to see broadening of those distributions. An additional point that we also noted in the simulations is as we get very close to the phase separation boundary, we start to see these rolling aggregates. You might have a couple in two aggregates, say in the whole simulation, and they roll around the simulation cell. Where we get this, we get these heavy-tailed distributions, and these are representing real micro separations. And these were quite a clear signature that we saw in a number of the simulations. Finally, the phase-separated case. In this case, we expect the material to largely be localized into a subset of those subcells, with only a few molecules then exchanging with the rest of the material in the simulation. And this we expect to produce a strongly bimodal distribution that we would be able to recognize quite easily. So utilizing this methodology, we were able to summarize the results into distributions and be able to draw some insights from those directly. In order to predict the phase boundary, however, we now needed to quantify these distributions and use those to define where we believe the phase boundary to be. So we went to the literature and we began to have a look through various different metrics that could potentially do this. And we came across one known as the bimodal coefficient. Physical sciences, and it's shown some pretty promising results. The coefficient itself is generated from the third and fourth moments of the distribution. So this is the skew, representing the side to which the tail of the distribution is skewed, and the kurtosis representing the peakedness of the distribution. So this is how sharp is the peak, or how flattened and bread tin-like is the distribution. In completely precise terms, the coefficient actually uses the excess kurtosis, a normal distribution, which would have a kurtosis value of 3, hence the -3 on the right-hand side of that top equation. Bimodal coefficient itself is then calculated using the equation in the lower right. The metric itself has a strict cutoff at 0.6, so it takes values between 0 and 1. Below 0.6, you would find that the distribution tends towards unimodality. Above this, it would tend towards bimodality. We also suggested that we could set an additional cutoff around 0.45, and that this would likely capture the regions where we'd see those microphase separations. So what did the results look like for predicting these phase boundaries? Well, taking the DPD case first. So in these cases, we have the experimental line shown with the dashed purple, mixed points represented by green dots, and red points representing phase separated regions. The orange points represent these ambiguous micro separations. So you can see here that the simulation is predicting a phase boundary. However, that boundary looks to be too low on the diagram. So it seems to predict that mixing would continue for approximately 10% less methanol than 10% less methanol than actually occurs experimentally. Still, looking at this, we can see that a phase separation is occurring. It's just that perhaps the sampling itself is not able to provide us with the precise, crisp details of where that boundary exactly lies. One thought we did have for this is that we've now mixed two parameter sets, one from the original parameterization by Anderson et al. And one that we have parameterized ourselves. And perhaps there's something that we could do to improve this slightly by actually taking the whole set and parameterizing it in a self-consistent manner. For the molecular dynamics, so in this diagram, we see that the coloring has the same meaning, but here we see that the diagram much more closely matches the. So there's a couple of points that are slightly below the boundary, which is still rated as mixed, but typically these are fairly close to the boundary for transitioning. So remembering 0.45, so the numbers next to it represent the metrics value. So 0.45 and 0.42 are getting quite close to being in the ambiguous region. So these are moving towards being in that micro-separated state. And you can see that there's in orange in the top right-hand corner. So this is actually sat right on the border. So it's been rounded to 0.45, but in actual fact it would be slightly above 0.45, hence why it's ambiguous. But that one's actually sat right on the boundary. So it looks like, looking at those values, we're actually getting something that's representing quite close to showing us where that boundary would actually be occurring. So we were quite pleased with that result. It showed that this simulation method was able to actually capture quite a bit of the position of that boundary. So in summary, what I've shown you so far is that we can utilize molecular dynamics and distributed particle dynamics for simulating ternary phase diagrams. In both cases, we can see a clear boundary forming where there is a clear phase separated region. However, it looks like the dissipative particle dynamics in this particular case hasn't managed to capture the boundary's accuracy to the same extent as the molecular dynamics. This might be to be expected, as the molecular dynamics has a much finer degree of control over those chemical functionalities and interactions. And also we suspect we may be able to improve the DPD results with a full self-consistent parameterization of a given potential set. I'll now move on to the second case, which is considering a binary phase chemical system. And we're also considering an additional complexity, which is the temperature dependence. So we start at the same point. We began by considering experimental data and looking for something that could act as a benchmark. In this particular project, we wanted to look for something that was slightly more complex chemically than the molecules we'd looked at previously. And we also wanted to include temperature to see if we could capture this effect. In this project, we're working exclusively with dissipative particle dynamics, and we wanted to proceed purely with that as the simulation method. We found the work of Chang et al., which actually provided several phase diagrams, which seem quite well validated on a single diagram. We decided to follow the boundary, which is outlined in red, principally because we had the potential parameters that were required or could generate them from previously defined sets. Great. So, okay, these two molecules are both much generated these using 2D representations of the molecule and generating 3D coordinates from those using our RDKit. We then used our own in-house codes in order to coarse grain the models, which again you can see using those red translucent beads, which show where that coarse graining is occurring. So now we had our experimental data, our potentials, and our different systems and molecules that we could use as our input. What we needed was a metric. Now, in this phase diagram itself, there's an additional complexity in that if you look to the top left, you can see the L representing a liquid, and the lower right saying L plus S, which is liquid plus solid. So in this particular case, the octadecane would actually be a solid below 27 to 28 degrees C. And so we decided that we needed a parameter that would be able to capture this. So we turned to liquid crystal research in order to find such a parameter. And we located the isotropic pneumatic order parameter. And this is a global order metric, which essentially represents the degree of alignment or misalignment of a given set of molecules. So the method functions by defining a so-called global director, which is represented by the vertical black arrows in the lower diagram. Direction of this subset of molecules that are selected. In this case, this is all of the octadecane molecules in the system. And then it computes the deviation of the molecules in that system from this global director. If all of the molecules align perfectly, then we have a value of 1. If they misalign, we have a value of 0. And reality says that most of the systems will sit somewhere in between these two bounds. Given that the octadecane isn't going to form a crystal in this case, we weren't expecting to see the extreme end towards 1, but we would expect to see a value above 0.45, which we would consider to be representing some kind of solid amorphous waxy structure for the octadecane. So now that we had the order metric as well, we were able to encode our workflow and run our first set of simulations. We did so with 3 different temperatures and 3 different concentrations. The diagram that we produced is as follows. So the red dash curve here is approximately representing the position of the experimental boundary, and the 9 sampling points, which are represented by the dots, are colored by the value of the pneumatic order parameter. Now, hopefully you're all squinting at your screen, slightly looking for a difference in the color. There isn't one. So in this first run, we actually found that the simulation had very little sensitivity across the concentration or temperature. And as a result, we couldn't see any transition at all. This was slightly disappointing. But we took the simulations away and we analyzed them and began to have a look at what was occurring inside them. And noting the prefactor here of k over 2, this k is representing a spring constant of how difficult it is to bend and stretch this angle. We decided to try out a few different values for this k. So this was effectively a manual parameterization. Typically, we set that to a value of 5, which leaves the angular variation quite free. And we decided to try values from 20 up to 50 in steps of 10. We ran these simulations at a single temperature, so just at 25 degrees C, to see what would happen across the concentration variation. Okay, and in blue you have the triglyceride. You can see now quite clearly, looking at the orange diagram, going from the low concentration where the triglyceride quite small, seeing any order there. In the center, we can see that there's actually some, hopefully you can make out in the diagram, that there is some local order, but there's no global order to the octadecane. On the very far right of the diagram, though, we can now see there's some real global order to that octadecane and some triglyceride bubbles effectively sat inside it. So we're quite pleased with this. Changing the angular potential seemed to have allowed us now to discern a liquid to solid transition within dissipative particle dynamics across a concentration range. We then began to think about this temperature variation again. And given that in the first diagram, we'd seen no real sensitivity to this, and it was a liquid in all cases, we were now wondering if, having increased that value, if we'd actually effectively made it unmeltable within the simulation. So we decided to test this out as well. We ran the original 9 sampling points, and then we ran an additional 9 sampling points, which are all at a higher temperature. So now we were going from 20 up to about 60 degrees C. The results are as follows. So now we have the diagram as I showed you before, lower side, plus the additional 9 sampling points on the top. And then on the left-hand side, we have the snapshots of the final step of each of those simulations. Just to confuse matters, I've actually changed the coloring scheme. So the green here is the octadecane and is the tricropyllene. Right, look at the color dots that there is in the color. So on this diagram, dark colors are representing low values of the order parameter, and higher values are represented by brighter colors. So looking at that right-hand column again, we can now see that the brightest color is at the bottom right, and it moves progressively towards the darker colors, which suggests we're actually capturing some of the trend towards the transition. I said that octadecane melts around 28 degrees C, which is roughly where the red dotted line meets the axis on the diagram. So looking at the points there, so just above 30 degrees C, we can see that there's still a slight sort of dark green hue to the point, suggesting that there's still perhaps some solid character. Above that, though, we see that it clearly turns into a much darker color, suggesting that the transition has occurred in that region. That suggests that we've got a 12 to 13 degree error, something in that range, in terms of the melting point of the octadecane. This isn't too bad. This is roughly in line with some other simulation methodologies and force fields in molecular dynamics, but does show that we're able to get the transition. To quantify this slightly more, this table shows the actual numerical values. So the pneumatic order parameter shown in the very center column, and the temperature is given and concentrations are given in the first 2 columns. So every third one down shows that right-hand column. So we can see at the low temperature, 20 degrees C, have a value of 0.63, 25 degrees C, a value of 0.57, suggesting these are both solid. And then at 30 degrees C, we see 0.43. As I said, we expected solids to be more or less above 0.5. So this suggests that perhaps we are actually capturing the transition slightly earlier than that 40 degree sampling point, and perhaps the error is slightly lower than we were estimating from those sampling points. So in summary, we demonstrated the modeling of a binary chemical system over temperature using dissipative particle dynamics. We've shown that the angular intramolecular potential is important in terms of representing that structuring and ordering of the system, and that it can be tuned to match to these experimental properties. We suspect that this won't be a particularly transferable parameter and perhaps not useful directly in other simulations, particularly not where the chemistries move outside of the families that were used here. But it may be something that could be tuned on a specific basis that could be useful for certain systems that are routinely screened. In this case, we just applied a simple parameterization of selecting a standard step and testing a variety of different parameters, and this has worked out. It may be that we could apply similar parameterization methods to those that we use in the AIJ cases in the conservative potential to more systematically tune these, but this would be additional work and then further work to consider whether these may in fact be transferable or not to other systems. So in summary, demonstrated the ability to explore phase diagrams using 2 different simulation methods, molecular dynamics and dissipative particle dynamics. In both cases, we've estimated the position of phase boundaries using a set of different metrics and measures. We've compared the 2 simulation methods, and we've also shown how we can potentially parameterize in some dependence on other physical changes, such as the temperature. With that, I would like to thank you for your attention and my colleagues who've worked on the phase projects with me. and I'll look forward to any questions. Thank you very much.
Thank you, James. So I would like to ask our listeners to ask any questions using both chats. James, I have a few questions. The first question is just how long does it take to run these simulations in order to observe phase separation?
Mm-hmm. So in the so that's a good question. So in the molecular dynamics cases, the whole workflow, which would, I've forgotten how many number of points we're actually on there now, but to run all of those simulations was taking a period of several days. So this is probably 3 to 4 days, I would estimate on average. And that's assuming that you have the immediate resources to run those simulations. Queue time would be in addition. Distributed particle dynamics is slightly faster, mainly due to the fact that it's a bit faster to equilibrate. So it's probably on the order of 3 days.
Okay. But in terms of, I think, well, I don't know how to specify time in lengths, but if you use GROMACS, you just put, let's say, 15 nanoseconds, and then you run over this time. And yes, this 15 nanoseconds might take like 1 week or a month. So in terms of these nanoseconds, how long does it take?
Okay. So they actually differ depending on the sampling points. So we look for equilibration metrics, and where systems aren't equilibrated, we then extend the runs until they do reach the values for those metrics. So I would say on average, there's probably about 4 to 5 million time steps, 4 to 5 million femtoseconds. In the molecular dynamics, DPD is harder to estimate in that time scale because it doesn't translate directly through sort of analytic formulae into a sort of real time scale. And this comes down to part of an artifact of the potential. So the potential is soft core, which means that the molecules can move through one another at a relatively high energy state. This is part of the reason why it equilibrates faster, but it also means that you can't get a realistic time scale out of it without having a known system that you can measure it relative to. So it's hard to say exactly how long the DPD simulations are actually representing in terms of real time.
Thank you. We have a few questions from YouTube. So a question from Philip Kemp from University of Edinburgh. In three component simulations, the microphase separations give large interfaces between phases. Is that a kinetic artifact or a real thermodynamic effect?
Sorry, the line broke up at the end of that question. Could you possibly read that again?
Yes, sure. In three component simulations, the microphase separations give large interfaces between phases. Is that a kinetic artifact or a real thermodynamic effect?
Sorry, I'm still not catching the question. Is there somewhere I can read that?
Just give me a second, then. Please use your chat.
Thank you.
And go to YouTube. Or you can read in Zoom.
Yeah, I've just read it. Thank you. So that's a good question. To be honest, it's not something we looked at directly. The systems themselves will have reached some kind of equilibrated state in that form. So I would suggest from that, on that basis, that there is some stability there. But whether it's an artifact of the kinetics or an actual thermodynamic stable state, I would have to actually investigate that. We haven't done that work, but it's a good question.
And another question from Philip Kemp as well.
So I'm assuming that's in terms of the subcells that you're referring. So the question: have you tried cuboidal boxes with small planar interfaces? So the subcells that we defined are cuboidal boxes, and they were defined to encompass a certain proportion of the system, so that we could we were making sure that we weren't generating very, very tiny boxes, which couldn't really capture the aggregates of the system that would be forming. In the molecular dynamics cases, those boxes were, the simulation itself was much smaller than in the DPD case, simply for the cost of the simulations. So in that case, the sampling would have been slightly, it would have been less than in the DPD case.
All right. I think we don't have questions anymore.
Okay.
So I would like to thank James for this very interesting presentation. And next time, just a second, James.
No problem.
Continuing with questions. And some comment from Philip Kemp. Question 2 is related to question 1. If you started with 2 separated phases with one small interface, would they go back to a microphase separated state?
I see. Okay. So again, that's a very good question, and it's not a simulation that I've run, so I couldn't really comment on what the output of that would be, but that would be an interesting thing to do, but we haven't done that, so I couldn't answer the question.
All right. So I would like to ask our listeners if they have more questions, then please ask. Probably we don't have questions anymore. So I would like once more time to say thank you, James, for your presentation.